Hello everyone and welcome to today's virtual panel. On behalf of Drug Target Review and Eurofins Discover X, I'd like to thank you all for attending today. I'm your host, Victoria Rees, Editor of Drug Target Review. Today's speakers will be Ramsey Connor, Senior Scientist, Project Director, RSO at Catalent Pharma Solutions, Pete Brogley, Cell-Based Assay Group Lead at KBI Biopharma, and Marcus Ruka, Managing Director at Vela Laboratories. Today's moderator and discussion chair will be Gaurav Agrawal, Associate Director of Market Development at Eurofins Discover X. I'll be passing over to them soon, where they'll have a fantastic discussion, and after that discussion, we'll be moving on to a live question and answer session where you can pose your questions to all of today's speakers. Please remember that you can submit your questions at any point during the virtual panel using the questions panel, which is situated in the menu on the right. But without any further hesitation, may I pass over to Gaurav to lead today's panel.
Thank you, Victoria. I hope everybody can hear me. And thank you to DTR for organizing this. Welcome, everyone. I'm Gaurav Agrawal, Associate Director of Market Development at Eurofins Discover X. My role here is to support our cell-based assay products, and I closely work with our partner CROs and CDMOs when transferring our assays for potential auto-release testing for our clients. I'll be your moderator today, as Victoria mentioned, for this event. And with me, I have a great panel of industry experts from leading CROs and CDMOs, and whom I will now request to introduce themselves. Thank you. Ramsey, would you like to start with yourself?
Sure, Gaurav, I'll go first. Thank you. Good evening, or good morning, good afternoon to everybody out there. My name is Ramsey Connor. I am a senior scientist in the biologics department at Catalent Pharma Solutions in Mooresville. I have been here working at this site for 14 years. And just to go over just a few things that we support here at the Mooresville site, we perform pre-formulation screening, formulation method development, stability storage testing, structural chemistry, characterization, bioassay, extractable leachables, metals testing, compendia, as well as inhalation and commercial manufacturing. So we are also a certified service provider of Eurofins Discover RX for cellular bioassays, and you can find us on their website. So look forward to answering questions today.
Thank you, Ramsey. Let's have, why don't you go ahead, Pete?
Hi, everyone. My name is Pete Brogley. I'm the cell-based assay group leader here at KBI Biopharma in Durham, North Carolina. We're a full-service CDMO. We support manufacturing, stability, analytical and formulation testing, pre-formulation testing, process development, and analytical development. I've been with the company for about 4 years now, and we support bioassay testing for stability, batch release, formulation development, and all sorts of potency testing. And we are also a certified provider for DiscoverX bioassay methods. We can also be found on the DiscoverX website, and I look forward to speaking with everyone today.
Excellent. Thank you, Pete. Marcus?
Hello, everybody. Can you hear me? Hello,
Yes.
welcome. My name is Marcus Ruka. I'm Managing Director of Vela Labs. We are a GMP and GLP certified company in Vienna. I have a degree in medical and pharmaceutical biotechnology. I'm working for this company for 15 years now. I started as a senior technician in the quality control, headed the lab for many years, and started now the business development. And as my speakers before, we're also a certified partner of DiscoverX, and we are pretty proud of it, and hopefully get your question answered quite quickly. Yeah, thank you.
Perfect. Thank you, Marcus, and thank you everybody for introducing yourselves. So yeah, we have a great panel for you to send your questions, and I will start with some poll questions actually for you. And these poll questions will have multiple choices. So you have about 30 seconds to click for your answers. And probably, yeah, first poll question is already here. So let me read it for you. So what has been your most recent experience using a commercially available cell-based assay? And the options are a bad experience, good experience, it's been excellent, or you have not used an assay from a commercial provider. You will have about, I think, 20 seconds to click for your answer. Please go ahead and do so. Okay, I think we can move to the second poll question now. Okay, here's the second poll question. So what challenges were you able to overcome when using a cell-based assay from a commercial provider? This should be a simple one. Here are the choices for you: assay reproducibility, assay robustness, mechanism of action, or time saving. So you have again 20 seconds to click for your answer. Okay, let's go to the last poll question now. So here's the last poll question. How soon do you start looking for a cell-based assay, a potency assay for your biologic or for your cell and gene therapy drug candidate in your pipeline? This is an important question, and we'll be discussing these questions at the end of the session. And here are the choices for you. So you start looking for an assay when you're at the preclinical stage, at phase 2 or phase 3. Please click your answer to this question. You have 20 seconds to do that. Okay, I think we are, so have these poll questions set aside, and we will review your responses and also bring these questions to the panel as well towards the end of the webinar to discuss your responses, as well as get some feedback from the panel. So now I would like to move to the next session of the roundtable, and we'll request the panelists to turn on their camera so we can start the roundtable discussion now. Thank you. Okay, so how we have structured this is we have a set of questions that we have put together, and I'll be having these questions open to the panel and see to get feedback. And these questions are again very relevant for potency testing, for your biologic drugs, for your biosimilars when they go out, for market release through QC lot release testing. So let me go to the first question here and open it to the panel. So the question is, what are the most common challenges you see with cell-based potency assays when setting up lot release testing for your clients? So I'll start this one and say, Pete, would you like to take this one?
Sure, absolutely. And it's a fantastic question. Some of the common challenges we encounter with our cell-based potency assays include defining the most appropriate cell growth conditions for the accurate, reliable, and consistent data generation. And that takes into account any of the different portions of the assay, whether or not you're looking at cell proliferation, cell viability, cytokine release, or what have you. Additionally, generation stability of a titration curve that will accurately determine the potency of your test sample throughout the range of the assay, and I'm talking 50 to 150, is imperative to define early on. So that way, if you have to look at other variables, including cell density or other such variables, having that as quickly as possible is imperative.
Very interesting points. I will see if somebody else from the panel have additional thoughts. Ramsey, what do you think?
Sure.
Challenges.
Pete gave a lot of great answers there, and I'll try not to reiterate what he said. But I'm gonna start off with the timelines. You know, one of the things you do when you kick off a project is you have a certain timeline expectations, and they're customer specific. You know, we like to be reasonable with our timelines, but sometimes our clients, you know, come in with a rush timeline. So that could be one of the challenges that we have to accommodate. Next is critical reagents. That's something that we have to specify in our methods, regulatory wise these days. And, you know, since we're talking about cell-based assays, you know, the cell bank is the most important critical reagent in this particular, you know, aspect. But you also have, you know, your reference material, and then you can have minor components such as, you know, your serum or your agonist, as well as stimulatory chemicals within your cell-based assay. You have to, a lot of times those are lot specific, and you have to qualify them based on what lots you use. But another thing you have to look at is assay interference, and these are things you have to consider when developing assays. You might have to adjust your media components or solution used for reconstitution, as well as, you know, mixing of other materials, assay plates, edge effect, things like that. So it's a lot of components that create challenges.
Perfect. Very. I'll say we have seen not in every program, but I'll say some of the things what you both mentioned in every other program that could be challenged, and I know the ways to overcome those challenges and kind of move with that. Any other thoughts from the panel, additional ones, any challenges that you have in mind other than what Marcus and Pete spoke, sorry, Pete and Ramsey spoke about?
I think they're quite great answers. Thank you.
Okay. Perfect. So thank you both Ramsey and Pete for the responses. I'll move to the next question. So the next is, what would be the key advantages of using, now this is the key to what we do here. So what are the key advantages of using a ready-to-use assay versus a continuous culture cell line format? Example, you know, what do you think? It saves time, increases assay robustness, reproducibility, and so on. So what are your thoughts on using an assay-ready format versus a cell line, which has been kind of established in the field that people use cell lines from ages, and now we are kind of, you know, being more sophisticated, trying to kind of move towards a ready-to-use format. So what are the advantages according to kind of your experience that you have seen while using ready-to-use? And again, open to the panel, and yeah, please.
Maybe I can take this one because we're using a lot of assets on various projects. So we really appreciate the availability of these ready-to-use cells, which can be bought as a reagent from Discoverex. And it also reduces then the time for the cell characterization. Like, for example, we're doing population doubling, growth curves, viability curves, receptor density, and so on. And for us, it would save up to 12 weeks of generating any working or master cell bank. And additionally, you have the advantage of having really high specific cells with a luminescent readout, which increases for some application also the sensitivity of the assays and also saves material. Furthermore, the lot-to-lot consistency of this type of kits and cells also saves time when you think about this life cycle management of methods, and also you reduce the time of, for example, FCS briefing or something like this, which comes along with the assay reagent list. Yeah.
Very nice, very interesting points, Marcus. Yeah, I'll let Pete and Ramsey see if they would like to weigh in.
Yeah, I like to weigh in on that. Those are good points, Marcus. Very good. You know, with my experience using a ready-to-use assay versus the traditional cell banking route, I felt like it was a game changer. You know, Marcus mentioned time. Definitely, you save time not having to grow up the cells. I mean, you have to consider the passage limits that certain cells have. You know, having to start at, you know, passage 4 or greater, versus, you know, with the Thaw and Go platform, you just start. You know, you throw your cells and you add your components to the assay, which increases efficiency. I mean, the efficiency is much improved. Another thing is robustness / reproducibility. I've seen great, reproducibility in terms of the variability of ready-to-use assays. The CVs are a lot cleaner in between replicates. The signal-to-noise ratio is very consistent, and it goes back to the time. I mean, that saves time. It saves rework. So you don't have to perform assays over and over again that fill acceptance criteria. And also procurement of materials. I think all of us had the challenge of procuring materials throughout the pandemic. And when you procure everything in the ready-to-go assay kit, then you don't have to worry about so many different components of the assay that you have to order and worry about back orders and things like that.
Very interesting points. And given what you said, Ramsey, is during COVID we have seen shortages on very basic needs that we have in the lab. But I will ask Pete, any additional thoughts?
Absolutely. And Ramsey and Marcus brought up some really great points as to the key advantages. And they hinted at the key point I'd like to bring up is the inherent variability using a continuous culture method. As you're passaging the cells, there are a number of variables that come up, including, as Ramsey discussed, passage number, but also trypsinization, centrifugation,
Yes.
and even environmental factors, including a CO2 variability or temperature variability. And if we're using a multiple cell line cell-based assay such as ADCCs, that inherent variability now just doubled. So using the ready-to-use kits really reduces the number of those variables that can impact the assay and the assay performance.
Thank you, Pete. Those are, I will say, very, very close to us, given we have a large portfolio for cell lines. But I mean, we are constantly moving towards developing more ready-to-use formats for potency testing. And some of the points that you mentioned, we do see timelines being expedited while we are transferring ready-to-use formats to these programs versus cell lines, which kind of, it's I will say it's not something that we say it's not going to happen, or it will be more hurdle prone. But we have seen much smoother transfers when we are doing with ready-to-use formats versus the cell lines. So really, really nice points. Thank you. So let me go to the third question here. So the question I have is, what are the most typical reasons that a cell-based potency assay fails? Now, this is a good one. During validation and any early signs to avoid such assays, which is kind of an important one. Where would you say an assay kind of shows you signs that this is not something that you should move forward with, or something early that you see, and probably help people listening in to be careful while they're selecting assays for their programs, if they can see something early on, and probably think or consider other means or other assays or alternative assays for their programs. So again, open to the panel, and Ramsey, why don't you take this one?
All right, this is something I've experienced and feel very passionate about. When dealing with clients, dealing with our customers, it is very important to encourage them not to skip robustness testing. When I say that, I mean, you must test lot-to-lot variability of reagents. Test your ability to expand your incubation times, your instrument. Don't be so quick to be reliant on one type of instrument. Make sure you do some instrument to instrument analysis, because you might come into a situation where your instrument goes down and you need to have another instrument to perform your bioassay. Leading into that is improper pre-validation. I look at pre-validation as a mock opportunity to run your validation, right? To opportunity to test your acceptance criteria, and also look at how things are faring against the protocol specification, which is normally provided by the customer. A smooth pre-validation, which tests a variety of components, usually means you're going to have a successful validation. And don't be so accepting of untested changes that are recommended by your clients or even yourself. If you're doing internal work, don't be so inclined to incorporate changes after you've already performed your pre-validation to your validation that have been untested, because usually that is an opportunity that's going to create mistakes and pains in the future.
Very interesting. Thank you, Ramzi. Good points. Anybody else on the panel?
Sure. So the points are well taken, and you said there were quite a lot of things. And I see also Bioassays like a long-lasting relationship, like a marriage or something. You need to invest in time and in your methods to have a really good outcome. I would like to bring another, let's say, view on this a little bit. So if you're a service provider, GP certified provider, and need to do the batch release testing and validate these assays, of course there are assays like ADCCs or something like this, which are prone to fail, which are quite complex. You need a lot of different kind of reagents and so on. And what we have experienced and saw is that, of course, you once in the life cycle of the product, you need to show that the real ADCC works, so the real mode of action is done. But what we have done then later on and discussed with the clients was really to cross-check this with a reporter gene assay and find the parallel, like potency and so on. And then you moved or reduce the risk of having this complex assay to a reporter gene outline, and have therefore a cross-check and so on, and can overcome this risk of failing during the validation at the end. Yeah.
Interesting points, Marcus. Thank you. I mean, from DiscoverX point of view, I mean, we have been in this space and, having 1,000+ assays in our portfolio. It's kind of an important point what we release and what the QCs that we do before we release it. And I think I agree with the point that you mentioned is there are early signs. There are signs that tell us that this assay may be an issue, and sometimes the assay may look really nice, really good, kind of nice window, nice potency, and everything. But I mean, at times, as Ramzi was saying, the robustness is very important. Is the assay benchmarking every time it's performing? And it's kind of proof is in the pudding, kind of say, unless you don't do it, you don't kind of run those assays a number of times. People come and ask us, oh, if we implement this assay, how it will happen. I was like, you need to do it to understand. But I think if an assay looks good, if it has good assay window, things like that, I mean, those are nice places, I will say, that assures you that this is a good starting point to do it. And that's why we kind of really recommend performing a qualification work, pre-validation work, before you kind of really think of an assay that this is it. So I mean, there are definitely checks and balances needed to be able to determine that this is the assay and this is going to be with you for your drug candidate for 10-15 years to come till your program goes on. So very nice points. Thank you to the panel. I will go to the fourth question on my list, and this is the final question for the discussion, and I'll then come to the poll. So the fourth question is, of course, I will ask this question from DiscoverX point of view. So basically, what will be your response for a key feature of DiscoverX assays that helped you, progressing a client's project, performing it in the lab, and things like that? What that would be that feature in a DiscoverX assay that you have used in your lab? Anything that critical comes to your mind really kind of help you say that, okay, this is the assay that kind of did this for me. So what that would be for you? And Marcus, why don't you?
There are many popping up in my head, and I do not know where to start actually. So at least we appreciate the very good technical support, right? If we use one of these assays, there is always at least a very good protocol, well-written protocol. You have sometimes access to your qualification data, which is also quite helpful for any kind of troubleshooting. You have so QC checked your applications and so on. And what makes it for me so special is, and it was quite stunning right from the beginning, was that you have an quite extensive list of available assays, right? So almost every solution was somehow found in the catalog, and if not, you have some access to a connection to the development team, which uses your platform basically to generate the tailor-made layout for the respective mode of action. So I really appreciate this collaborative work together with you.
Thank you, Marcus. Thank you. It's great to hear your good experience with that, and of course technical support team is important. I think that kind of goes to the heart of things, that how we support our assays is kind of very important for us to be able to support what we transfer to a different site outside DiscoverX. Any other thoughts from the panel? How about Pete? What do you think? You have used our assays. What has been your experience? What kind of stands out for you?
Great question. I just want to echo Marcus's sentiment about the usefulness of the technical support team. My team has, utilized a number of DiscoverX assays, and as we're doing, we may run into potential issues or concerns or thoughts, and we get almost an immediate response from the technical support team, and we're able to obtain some guidance, obtain some information moving forward. I know one experience right off the top of my head where we received a response within 2 hours. And the representative asked to look at some data. We shared it with him, and he was able to provide an answer almost immediately. So, oh, here's what's going on. They had seen that before, and it was almost an immediate solution. And in addition, the amount of technical documentation that's available just to really understand how to utilize the cells, how to manage the cells, and work with them definitely gives us a leg up as we're initiating a program.
Thank you, Pete. Very encouraging to hear that. Ramsey, anything that comes to your mind using DiscoverX assays?
Yeah, my experience is very similar to Marcus and Pete here. So I'm gonna just kind of piggyback on what they're saying. But by starting with the technical support, one of the things I liked about using the commercially based assay from DiscoverRx is that a great level of development has already gone into the assay. And a lot of the kits that we've used have been agonist-based response kits. And then we would introduce an antagonist in there, something like a monoclonal antibody that would prevent the response of the agonist. So, so you have your own challenges by doing that, by introducing a antagonist, because maybe your dose response level changes a little bit, your signal to noise ratio may change. As well as some other things, maybe some assay interference. However, I've had a great experience with the optimization, being that I inserted an antagonist, and when I saw troubles, maybe with some lot-to-lot variability, I immediately contacted technical support, and DiscoverRx was able to help. But what I found in the past is that with these assays, it's very easy to train analysts, easy to procure the reagents, and the optimization and pre-val was very speedy, regardless of the challenges that we ran into.
Very nice to hear that, Ramsey. And I'll convey the thoughts that you have shared with technical support team, and I'm glad that we are able to help. And I know concerns, as Pete was saying, issues, they are sometimes they're part of the thing. I mean, this part of the process that when you, and Ramsey, what you said is, we develop an assay for an agonist, and then you are going to use it for antagonist, and that's anticipated that you will do that. But sometimes, of course, antagonists come from your clients that we have not access to, we have not run those, and you are basically the one who is kind of discovering that on yourself, how the assay performs. So this is kind of the dialed in part on some of the optimization that you will do at your end. But glad to know that we were able to at least share our experiences with you and kind of help throughout the way on whatever we could in optimizing those assets. That's kind of great to hear. And this is exactly what we strive for too, is when we transfer, there are moving parts, because it's kind of, it's a kit which has multiple ingredients in there that we have optimized and kind of, put together in a way that it's easy to run. But again, ingredients like antagonist mode and things like that can bring in some challenges and some optimizations that you will need to do at your end when you're looking at the assay. But I mean, our technical support is here to help with those regards. So I will close this discussion, particularly on the questions here, and I think I will probably say, or comfortable to say, that these are some really great insights that we have received from the panel, and challenges and some of the things that you need to see early on in the assay and things like that, that will probably help customers to be a little bit more, I will say, watchful on things if they are using an assay, if they're building an assay their own, or they're getting a commercial provider like DiscoverX to use the assay, so they know what to look for in the assay and what could be some of the important details that they need to kind of put their minds to when they're selecting this assay. So this is a very nice discussion on this panel. Now I'll go back and review the poll questions, and I know I have received some responses from the poll. It's a good number of responses here. So let's look at the first poll question and open it to the audience, open it to the panel to discuss the responses. So the first one was, what has been your most recent experience using a commercially available cell-based assay? And I see quite mixed response, at least for the last three. A good point that very little number of people have bad experience, and there is good, excellent, and some people have not tried commercial provider for their needs. So why don't I ask probably Marcus, what do you think of the poll here? What are your thoughts on trying commercial providers, and what has been your experience?
I'm sorry to hear that some of you had bad experience with reporter genesis. Must be quite a complex topic, actually, or maybe the window was not good enough. So sorry to hear that. It would be great maybe to see maybe some of the questions. So I have to say we had a recent project together. To my point of view, and this is real personal view, is it was quite a novel project where we had a let's say a replacement of existing assay, the Steelman Poly test for FSH, which includes an in vivo rat model, and together with the clients there, we tried to reduce this animal testing for any kind of other assays, and therefore we collaborated together and used this platform to clone FSH receptors to the cells. And of course it was a tricky part because we hit the open window, because we need to, hormones are need quite a long a broad range of concentration to be tested and so on, but at the end we managed to find a quite good solutions, even if the subtypes, so FSH on this the subunits and so on, this were quite specific and robust and reliable. So to my experience, we always had very good experience, and yeah, thank you for the question, basically. Thank you.
Thank you, Marcus. Good points. Other thoughts, Pete?
Yeah, absolutely. And I'd like to echo Marcus's sentiment about, on the unfortunate of having bad experiences. I've definitely had experiences where I get something and it should work and then it doesn't, and everyone's just scratching their head. The thing I would advise people is that with commercial off the shelf products, is that whatever it is, you have to put it through its paces. You have to make sure it does what you want it to do. And there's some times where, unfortunately, if you get the product in-house and it doesn't immediately work, that there may be some additional development or some factors that need to be considered prior to saying yay or nay, the program is working.
Good, thank you. Ramzi, any additional thoughts?
I'm sorry, Gaurav, I got disconnected for a moment. Can you repeat the question?
Yeah, so the question that we are reviewing, the poll question, is what has been your most recent experience using a commercially available assay? And the poll was probably pretty divided in the last three, which I kind of read is a bad experience, having a good experience, an excellent experience, and have not been, have not used these assays. So 6% of people said that they had bad experience with assays, while good, excellent were around 30%. And then there are another 30% people who have not used these assays or any from commercial provider. So just wanted to gather your thoughts, Ramsey, on in general experience. I know you guys develop your own assays, but wanted to see if you have some thoughts on, how's the general experience getting an assay from a commercial provider.
Okay, well, was that 60 to 70% had a bad experience?
No, 6%. So yeah, thank you
Oh,
for checking
okay.
that. It's 6%.
6%. Gotcha. All right. All right, that falls in line with my experience there. My experience has been, nothing less than excellent. I mentioned before that, when we have used the commercially available assays that, we inserted antagonists most of the time. We've also used it where we substituted a different drug in there that acted in an agonist function. But, very, very easy to use, very reproducible. Not much adjustments necessary, or not much optimization, and that has contributed to the success of the sample testing for the client.
Perfect. Thank you for the panel. And I mean, I will say overall experience that I'm getting from the panel is mostly good experience using a commercial provider. So probably that will encourage some of the people who have not tried the assays from provider. So I mean, that will save a lot of time, and at times time is of essential value. So if you have an assay which is there, definitely, given the experience of the panel, probably a good idea trying an assay and see for yourself how you will like the assay. I'll now go to the second poll question and review what the responses are. Okay, here it is. So what challenges were you able to overcome when using cell-based assays from a commercial provider? And I see pretty nice response, actually. People have seen better assay reproducibility, getting a good robustness, or trying to say, okay, mechanism of action is also, people have been experiencing good results with that, and time saving. So I will say assay robustness is something that most of the people thought really was bought in from a commercial provider. So let me open to the panel and see what their experience is in terms of these 4 key points in getting an assay from a provider, and seeing, how did that help you with your programs. Let's
start.
Yeah, perfect.
I'll go ahead and start. Yeah, when I was looking at that poll, I immediately, saw the reproducibility. And, I remember a time where, we, during the optimization of the assay and going into pre-val, that we kind of overtuned the specifications for the assay. So we put acceptance criteria that was a little too specific, or a little too tight. And after doing some additional study, some additional pre-val, we ended up loosening up the criteria, because we saw that the assay was robust enough, and everything else outside of that criteria was reproducible, that we didn't need to include that specific acceptance criteria in the assay. So we took it out and everything was fine. We initially put that in to avoid out of spec investigations, which we've had great performance not having any OOSs, as we call them. So that was a little bit of my experience there.
Okay, interesting. Any additional thoughts from the panel?
Yeah, this is Pete. From my perspective, the assay reproducibility and time saving are definitely things that were overcome using a commercially available kit. As we use the kit, it kind of answers a lot of questions, or it takes a lot of the variables off the table because they're already solved by the kit, and we're able to focus on the more specific portions of the assay, and we're able to tailor the assay to the molecule of interest much quickly and be able to meet our clients' timelines in a much more quicker fashion and answer questions as quickly as possible.
Thank you, Pete. Marcus, anything you'd like to add?
Just repeating myself a little bit, so I really appreciate this lot-to-lot consistency here, which can help increasing this lifespan and life cycle management. But of course, if you have to meet certain requirements, it needs to be more optimized. Of course, if you, for example, have going from duplicates to triplicates or even more replicates just to reduce further CVs and so on. But yeah, these are very rare cases to my point of view, and the participants here, I think, gave a very great picture to this poll here, and I think this reflects the RGAs.
Perfect. Thank you very much. And very much aligned with everybody's response as a robustness and time savings and reproducibility is something that is expected, that you're getting an assay from a commercial provider, so they know what they're doing. That's the kind of expectation, and good that people have similar experiences as kind of we think when we develop our assays, exactly these are the things in our mind to improve when we provide our assays. So let's go to the third poll question, the last one that we have, and kind of an important one. Okay, so I think I like what I see as a response. The question was, how soon do you start looking for a cell-based potency assay for a biologic or a CGT drug candidate, which is cell and gene therapy drug candidate, in your pipeline? And overwhelmingly, people are looking right from the preclinical stage, 94%, 11% phase 2. There are phase 3, 6% of people do seek an assay at phase 3. Okay, let's open this one to the panel and see what are the thoughts. Anybody would like to take this one and share something in their mind?
I'll just open up and say I completely agree with that.
Okay.
I mean, you really can't start soon enough. So, and the preclinical stage, I think, is the best time to start looking for a potency assay.
Completely agree. We advise our clients to do it as quickly as possible. Just the sheer time it's taken to develop, identify it, and a cell-based potency assay. We have an assay that goes 11 days, and so obviously our assay throughput is quite limited. So having that as quickly as possible helps us meet the overall timelines of the client.
Yeah. The question was a little bit directed to also the gene and cell therapy products, which we started a little bit from a different side, not only because we're starting from the reporter gene assay side, to be honest, but having this in mind and how the question was set is that we also experienced that even regulatory authorities ask more and more often earlier in the product development to come up with some kind of an assay. And of course, if it doesn't really show the total, let's say, mode of action, it's just a reporter gene assay, right? It really helps you to characterize your product quite early. And I think this is also quite beneficial if you have data like this and show it in the scientific advice meetings or any other regulatory meetings. Yeah.
Perfect. So I think overwhelming response from the audience as well as the panelists: start early, as early as possible, and that kind of is a golden advice to anybody looking for an assay. Start at the preclinical stage, when you are on the discovery stage, when you actually have the time to do so, to put a good assay in place. So this is an excellent point. Everybody is on board. So thank you for reviewing with me the poll questions. And next, what we'll do is look at some of the questions that we have received from the audience. Let me go to the first question and read it for you. So given some of the upfront challenges that are discussed here, how soon, so this is something that we have seen that we are just asking this one: how soon would you suggest a client start looking for a qualified robust potency assay for a novel biologic? So I think this somewhere answers the question from the poll question, is how soon that you will ask them to start. But now the question is a little bit more for a potency assay for a biologic. So do we all agree that we still would like to say that even for a potency assay, what will be the right place to start? I mean, of course, people would like to do things differently in discovery. at that time they need a cell-based assay for a potency assay, what phase you would say that they should be start looking for an assay? Any quick thoughts from the panel? Say the advice remains the same, or there's specific thing would you like to tell the panel for a particular potency assay, any particular stage?
So I follow up the answer I gave so far. So, and I think it's also reflecting what Paul said. Giving the chance to someone who develops this assay needs to set in these protocols, and reporter gene assays give the advantages to have easy follow the protocols and already have a good head start, and therefore even if you may think bioassay at the start and so on, and this is a cost factor, I think it saves you time right at the further progress of the product. And to our experience, as I said, regulatory and scientific advice meetings are running much more smoother if you have this data in place already.
Interesting. So Marcus, I'll say that your advice will remain the same, that start early and start at the time when you have time in your hands. Is that what you agree with?
Of course, you need to do your homework first and see if the FUSCAM properties and so on meet your requirements. You have HPLC and so on. But what I say is that the bioassay itself may take some time to get a robust setup, to know what you can expect later on for the manufacturing side, right? So the manufacturing brings in some variability into your product as well, and all these things need to be covered then for your analytical side as well. And yeah, this is what my opinion.
Very nice. Thank you, Marcus. Any thoughts from Pete and Ramsey? Is the advice remain the same for you too?
Yeah, I would say, yeah, it pretty much remains the same. Early on, you're characterizing your product, and depending on depending on the progress of you characterizing your product, you want to go ahead and start getting in the back of your mind, how your product is going to perform in a biologic system. When you do these in vitro assays, that's the closest thing you're going to analyze a biological response versus the clinic or in vivo. So it's great to start searching for a system to get your product in as your product progresses through the phases.
Very nice. Thank you, Ramsey. And I'll move on to the second question from the panel. And let's say if we have kind of a quick take on this. So the question I have here is, what is the best practice to report the potency value? Is it average of 3 assay values or a single assay value enough, even if using a ready-to-use assay? So the audience is asking, what will be the best way to report a potency value? Is it an aggregate of 3, or is it a single assay? What do you guys think?
So this is Pete. Just to answer that pretty quickly, unfortunately, it's hard to give just a blanket answer as to what is the right way, because these methods are highly tailored to the different molecules and the different mechanisms of action. It's really hard to say, okay, always use 3 plates or always use 1 plate. What I would advise is to perform the development, perform pre-qualification, pre-validation testing, and then make a data-driven decision as to what is the best approach based on the data you've obtained.
Thank you, Pete.
To piggyback on Pete, I'll say it depends on how variable your assay is. I've had experience with assays that were very clean, where we only needed 1 reportable potency number, whereas I've had some other assays that showed a higher degree of variability, where we would use a 2-plate format, and then we would use the average potency of the 2 plates to create a single potency. And then I've also had experience with assays that use more than two. But from my lab experience, it's usually a 1 plate to 2 plate assay using either one reportable or the combination of 2 reportables to give the potency of the product.
Perfect. Thank you, Ramzi. I think this is something both you and Pete weighed in, and it depends. Basically, the answer is this: you need to kind of evaluate and see, and again, the variability of the assay, how consistent it is, things like that will determine the final kind of reportable potency you will put in from one or multiple assays. That will be. Another interesting question we have here is, and I think this is something that we get from the field as well. So to the panel, the question is, how has been your experience regarding the acceptability of the assays with regulatory agencies and filings? So in general, what's the experience for cell-based assays that either you use from DiscoverX or from other providers, or your own assays? What are the kind of a general feel for you? What are the challenges that you have seen from regulatory agencies when you are, or the client finally submits a package or data from from let's say the validation work, or from the potency testing work? What is something that you have seen? Would you like to share something with the audience?
So maybe I can start. So we do have some experience, or at least some experience of with our vendors. As I said, we showed once in the drug development that a real assay, robust or not, shows the proof of concept and shows also the mode of action and its potency application. And then we made a cross check or crossover to a reporter gene assays, which reflects basically the mode of action, like the DiscoverX platform with this path hunter assay. So you just put things together, there is a luminescent light starting. So what we have done, we made a scientific rationale why we should use this assay, and to our experience so far, and we have really customers from all over the world here, was quite widely accepted over the last years, and therefore I hope Ramzi and Pete have the same experience with that, but at least we have very good experience on that. Yeah.
Very nice. Thank you, Marcus. Any additional thoughts from Pete or Ramzi?
I will say this. Marcus made a lot of excellent points there. I would just like to add this, that the acceptability from regulatory agencies has been pretty good with accepting these type of assays that show a mechanism of action, inside the cell. Oftentimes, this potency assay is coupled though with like an ELISA-based binding assay to also show that aspect of what the product is doing. I have also experienced some feedback from regulatory asking about the availability of commercially provided assays, such as provided by DiscoverRx. What would be the consequence if your primary provider, such as Eurofins, or would go belly up or something? What is your plan? What is your backup plan? So those are things you have to consider. I'm thankful that that's never happened to me. But those are definitely questions that come up, and you have to have some type of backup plan.
Very nice. Yeah, go ahead, Marcus.
I don't know if Pete wants to say something as well. I don't want to cut him here, but I want to bring it from a different perspective as well. So we are working also on a clinical perspective, for example, neutralizing antibodies and so on. And I don't know if the question was related to that. It was just, I think, related to what the regulatory says. And we have very good experience and even more better experience if we use it for clinical neutralization assays and so on, that they even more capable because they are highly specific. The receptors are more robust and so on. Lot consistency. And if you think about the clinical life cycle or the time how clinic is run, we had very good experience on that part. If you want to see them from this point of view, yeah.
No, of course it adds a lot of value, Marcus, because the two side of things. You do potency assays, and of course you are looking for NAB and anti-drug antibodies. So thank you for adding that. You have seen good results or good acceptance from the regulatory agencies. So I think I will just kind of add to what Ramzi was saying. We do have a lot of mechanisms, Ramzi. If you're getting questions like these from the regulatory, we do go ahead and sign long-term supply agreements where satisfies the regulatory agency need to see that companies like DiscoverX will stand next to the product for years to come. So we do have those provisions. We have done that for companies. So definitely we'll be happy to work on that and provide those supporting things to you that will satisfy the agency that DiscoverX is there to stay. And if not, there are risk mitigation strategies that we put in place. So the program is not affected because if anybody chooses to go in a different direction. So we do all those kind of arrangements for these programs, and they're very important to do that. And I think we have multiple other questions, but I think we are running out of time. So I will probably, what I'll do, maybe after the webinar, we will have those questions reviewed, and I'll get thoughts from you, and we'll respond back to the audience. So their answers, their questions are answered from the panel, but we'll just do it through email. And I will like to close and thank the panel and such a lively, such an interactive discussion, and the audience for being super nice, sending so many questions to us. And apologies, we cannot answer all of them, but we'll see to it that you have the responses from the panel after the webinar. And over to you, Victoria.
Fantastic. Thank you so much, Gaurav, and thanks to all of our panelists as well. That was an absolutely brilliant discussion, and well done for answering all of those questions. Thanks also to our audience for sending them all in. There was some really great discussion there. Unfortunately, though, that is all we have time for today, but please do join me in thanking Gaurav, Ramzi, Pete and Marcus for their time today. As you leave the session, a survey will appear on your screen asking you to rate this experience. If you could take a moment to provide your feedback, that would be very appreciated. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we'd greatly appreciate it. But now, on behalf of Drug Target Review and Eurofins Discover X, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Thanks so much.
Thank you.
Thank you.
Bye. Thank you so much.
Bye.